Hello, my name is Jessica Kerr and I'm from Scion. Today I'm going to talk to you about the Pikoura Protection Research Program. The Pikoura Protection Research Program was a multi-year MBIE research program that ran from 2015 to 2018 and will continue with Scion core funding until 2021. The aim of the program is to avoid future long-term pest management costs in urban, production and natural environments, developing new knowledge and tools in a layered approach that combines pathway risk assessment, surveillance and efficient early detection, new models and tools for eradication, and social and cultural co-design. The Pikoura Protection Research Program has multiple areas of research, and today I'm going to give you a summary of 5. Firstly, insect cyborg surveillance. During an insect incursion, speed of response is crucial. The faster the response, the greater the chance of success. Our research aims to actively find an insect incursion through a targeted biosecurity response tool. Many insects use chemical odours called pheromones to communicate. We have found a way to help us snoop in on the system and find them. Over the past 4 years, Scion has developed a portable electroantennogram, a lightweight tool utilising a male insect to track the pheromone of a female. The male insect is mounted in a device which can then be used for handheld active searching or placed on a UAV. In 2019, we completed 2 successful field trials in Canada with Natural Resources Canada's Great Lakes Research Centre, with key results showing gypsy moths could consistently detect insect pheromone within a wind tunnel at 30 metres and the field at 130 metres. The male moth's response is visible in the graph with the yellow boxes. We collected 676 recordings from 23 moths, a very large and complex data set that currently needs to be manually reviewed. To help improve and speed up this process, we're moving towards an automated system, and the project has been scoping a range of machine learning options for processing EAG data and delivering a prototype for gypsy moth to support our international collaboration. The machine learning algorithm picked for this type of data is a deep learning model. The model requires a training library of data so that it understands what is and isn't a response peak. We started with laboratory data, where responses were at known time intervals. The algorithm could clearly pick up the peaks after training in the bottom graph. We then moved into wind tunnel data, which requires annotation for training, visible in orange, selecting the start and the end of a response. Then running the trained model, it highlights the predicted responses in red. After training on a small subset of 3 moths, the model is close but has missed 2 responses. To improve this accuracy, we'll continue to build up a training library over the coming year, validating the model with this summer's field data coming in from Canada. Other new surveillance technology we're investigating is the use of traps equipped with cameras for targeted surveillance of insect pests. As part of a collaboration between Scion, Ag Research and the University of Canterbury, we've designed a product that will provide new ways to inform research. For example, at ports, we're planning to use these traps during and after a vessel docks to see if hitchhikers are jumping ship. Within forests, it will be used to monitor insect flight patterns. The traps are linked with the wireless Internet of Things communication network to send back basic information about the trap's performance. The project has been working on integrating machine learning for image recognition to differentiate between target insect species. The ultimate aim is to integrate these traps in the real-time Bayesian network on flight probabilities developed by Steve Pawson and collaborators, such that the sensor network can constantly improve itself. We're developing camera traps for real-time surveillance. Using machine learning to automate insect image identification, the trained image recognition model can currently differentiate between 3 forest insect species based on a small training data set. It's operating at 90% accuracy. Along the same lines of research, one of our teams has been scoping the design of a system that can implement image recognition for pre- and post-border images. The species I've highlighted for you are some of the most common found at our border. The project is aiming to work with inspectors to take an image of what is found on imported goods, applying image recognition to provide important information on different taxa of insects coming through our border. The next step for these projects is to gather training data of good quality images that can feed into the image recognition model. Watch this space because the science and technology is growing rapidly. Our next project is on sensor development for detecting plant diseases with volatile organic compounds. Volatile organic compounds, or VOCs, play crucial roles in plant defense and survival. Detection of unique VOCs emitted by plants has been suggested as a promising method to monitor fungal infections or insect infestations. Microbial emitted VOCs are presently used as markers to detect human disease, food spoilage, and mold in houses. The project aims to develop a lightweight drone-mountable pathogen sensor able to sense infections prior to visual symptoms or spore dispersal. The project hypothesizes that by monitoring VOCs emitted from trees, plants, or microbes that are markers for specific pathogen species will give us a tool that can be applied to different classes of pathogen or insect hosts at early stages of infection. Over the past year, the team has developed a VOC sensor based on carbon nanofibers and has tested the sensing performance using 2 volatile organic compounds typically produced by living organisms. The results show that the sensor can be used for VOC detection. Each peak in the graph is the sensor picking up the chemical. The team will focus their sensor development on the phytophthora responsible for cowrie dieback in the next year. Our atmospheric scientists have been researching a relatively new area of science called atmospheric Lagrangian coherent structures. Atmospheric transport of material by wind, including pests, follow preferred pathways. Lagrangian coherent structures are a way to visualize these pathways and their behavior, giving organized trajectories of winds, often called air bridges. The purpose of this study is to understand the behavior of LCS over New Zealand to optimize biosecurity surveillance sampling networks, to predict important events, and to stretch current science to understand LCS at a regional and local scale. Key findings so far from 11 case studies of trans-Tasman insect incursions show 100% are associated with the Lagrangian coherent structures. There is also a strong correlation with LCS air bridges and trans-Tasman smoke transport from last season's Australian bushfires. For each smoke event, the maps show the shape of the smoke in the atmosphere and the correlation with the air bridge. Material that is transported in the atmosphere will eventually become concentrated into these dark structures. As we see, the smoke in the cloud that is concentrated in the structures outlined in the red dashed lines. We believe this science enables us to predict long-distance wind dispersal of pests and pathogens into New Zealand, and possibly at smaller scales. Therefore, we can target eradication campaigns to where LCS predicts pests will spread. In summary, the Pikoura Protection Research Program is preventing damaging forest pests and pathogens from establishing in urban, production, and natural environments. Thank you for your support.